Okay, we've got the PowerPoint to record, not as crutch. Okay, so today we're going to talk about plant micropropagation. So what do you guys think that is?
Well, it was like the in vitro for plants, essentially.
Yeah, exactly. This is like in vitro. In vitro means like in the test tube. In vitro culture of plant tissue. And you can use this to generate essentially like clones from plants, okay? So let me first start with a preface to some developmental biology. We're going to do more developmental biology in the coming lectures, but let's just start with a little bit, which is going to help understand this lecture. So let me just first identify some terms. So growth, what do you guys think growth is?
Any time there's production of new cells.
It's actually, so that's actually why I point this out, is it's actually not a replication of cells. It's sort of like an increase in the size or volume. And so that implies that to grow, you don't necessarily need to replicate your cells. Okay, your cells could increase in size, or if you're an organism, maybe your extracellular stuff is growing in size. It's an increase in size or volume. Okay. Now you answer this question: what's cell differentiation? Or actually, I should say, another way to grow would be through replication, but that's different. So that would be like cell replication. Do we see how those are different? Yes. Okay, good. Okay, now as things grow, okay, there's a term called cell differentiation. Okay, what would that be? What would it mean for a cell to differentiate?
Kind of like start with like stem cells. And then
Yes, exactly. So that actually goes into the next thing is as organisms are growing, when they first start off, there's a term called totipotency. That means if you start off with sort of like the first cell, the first zygote of an organism, that cell has the potential to become, as it develops, any other type of cell. So if you're talking about animals, like the first zygote can eventually turn into a brain cell. It could turn into a skin cell. It could turn into like a muscle cell. And all these cells are different, okay? But as that process happens, that's called cell differentiation. That's a process you can't usually come back from, okay? So when you start off as an embryo, you're totipotent. That means you can turn into all these different types of cells. But as your cells differentiate, as you grow, as you go through your development, your cells essentially become what they're destined to be, and they can't usually go back to a totipotent cell state. So that's cell differentiation. Okay, what's morphogenesis? Again, these are like developmental kind of like preliminary terms. What would morphogenesis be?
Change in the beginning.
Change in the beginning. It's more like the development of like structures. So like organ system development. If you're a plant, like root development versus like bud development versus leaf development. Okay, that's kind of like morphogenesis. So as cells start to differentiate, the reason they're differentiating is because they're starting to build these morphological structures. So that's how you get like a brain, a skin, and muscle, different tissues, different organs. That's sort of the term morphogenesis. So these are sort of like three different terms that describe different processes that are related to growth in developmental biology. Okay, let's also distinguish between somatic versus germline. What is that? Have you guys heard these terms before?
That's somatic, like body cells.
Yeah, exactly. Body cells. Germline cells are the cells that pass on to the next generation, okay? So in your body, none of your cells will be inherited by your children except ones that constitute your sperm and your eggs, if you're female, or sperm if you're male, okay? So the germline are cells that get passed on to the next generation, and somatic cells are cells that die with you, okay? They inherit your fate. They die with you. Okay. So now one of the reasons this is important is this distinction is because when we start analyzing transgenics, how to make transgenics in plants or how to make transgenics in insects or animals, which of these cells do you think we want to target for transgenics? Body cells or germline cells?
Germline cells?
Yeah, germline cells precisely, usually because we want to establish a stable transgenic line that's going to be inherited for as long as we culture that organism. And the only cells that are going to be inherited are going to be those germline cells. So that's one reason why I sort of put this terminology here, because it's very important as we talk more about transgenics. Okay, so now that we know what differentiation is, what's dedifferentiation? So if cells progressively divide and become structures, what's the antithesis of that?
The ability to undifferentiate cells.
Yeah, exactly. So the ability to go back to a totipotent state. So back to totipotency. And that's atypical. In some, at least in animals, for sure, atypical. But in plants, if it's the knowledge of how to do this is sort of older. So in plants, you can take a somatic cell and you can treat it with some chemicals and it will revert back to a totipotent state. Okay, so you see how valuable this would be? You could take like a leaf or like a bud or a root, and you could treat that with some chemicals, and you could generate some cells that could then become a whole separate organism. And do you see how that's typically different from, I could not take a cell of your skin. Well, actually I can now, but I'll get into that in a second. In theory, I shouldn't be able to take like your eyeball and generate a whole new human being, right? Okay, but we actually did just learn how to take somatic cells in humans, and actually we can make what are called induced pluripotent stem cells. I won't talk about that much today, but essentially this is new knowledge. If you treat somatic cells, you can actually generate like a pluripotent stem cell. So this is sort of new knowledge. Okay, now you see how this relates to micropropagation of plants, because we can take these somatic cells, we can treat them with chemicals that bring them back to a totipotent state, and then we can generate new plants from these techniques. Okay, so now let's talk about the micropropagation. Why would you want to micropropagate plants? What are all your suggestions or thoughts about why would this be useful?
If you were trying to like, keep certain characteristics and you needed the very specific plant.
Selection, right, exactly. So there are a couple of examples of this. If we have sort of generated a novel phenotype by breeding and we want to keep selecting that and we want to clone that, we could take a piece of that plant and we could micropropagate it, and so we could sort of select for that phenotype. The other reason we would want to select is from the last lecture. What was that lecture based on?
Agrobacterial?
Yeah, creating transgenics. So if we create a transgenic plant, we want to generate a plant that then in the next generation sort of has that transgene throughout its entire body. So we could use this micropropagation as a means to generate clones for that particular transgene. So selection of clonal populations is super useful. Why else would we maybe use this? Any thoughts?
Large-scale production.
For sure, large-scale production. So if you are a horticulturalist or some type of person that's going to produce a bunch of plants for sale, you can use this to scale up production of those plants. Another reason you might want to use this is if, say, some plant that you care about gets infected, you could take a cell of that plant and you could put it in an in vitro environment and sort of cure it. If it was infected by a bacteria, you could cure it with antibiotics, or if it was infected with a virus, maybe you could take a piece of that plant that hasn't been infected yet, and you could cure it, and then you could generate a new plant that doesn't have the disease anymore. So you could use it for curing, okay? A lot of times, we talked about this before in the last lecture, a lot of plant biotechnology is in creating hybrids. And what's a characteristic of many hybrids?
They kind of have the best characteristics of the two parents.
Yes, but what's the downside?
They're sterile.
Yeah, usually they're sterile. So the question is, if you generate an awesome hybrid, how can you get more of that if they're sterile? A lot of times you can take little parts of that plant and you can micropropagate them. So it sort of circumvents the problems you generate with generating hybrids. I have one final reason, which is you could use this to generate mutants. So if you want to test, one of the issues with generating mutants is if you were going to generate a bunch of mutants and then compare them, so you're going to compare A to B to C to D, you would want these all to be the same background. That essentially means like you don't want any other genetic changes other than the thing that you're changing. So you can use micropropagation to generate clones, which then are only different in whatever you mutate, if that makes sense. Okay, so these are all reasons why we would want to do plant micropropagation. Okay, so this ability to do this was prefaced on the discovery of certain plant hormones. So the only reason we can do this is because we know a little bit about plant hormones. Did you have a question?
Okay, just my bad.
Okay, good. So what are hormones? Before I go into that, you guys know what hormones are?
They
Chemical signals.
They definitely can convey chemical signals. But how? How do they usually do that?
In humans in the blood.
They
I don't know about this.
You saying they float around in the blood? For sure. Usually, this is very common with hormones. Is they're usually like a chemical, okay, that usually circulates. So if you're a plant, maybe they'll be in your xylem and your phloem, and they'll get througout the whole plant. Or if you're animal, maybe they'll be in your veins and your arteries and your blood system. Or if you're an insect, maybe they'll circulate through your hemolymph. So all sort of organisms have these circulatory systems. And certain cells can produce these chemicals, okay, so they get into the blood, and their chemical composition will vary, so I'm not gonna, there's no like basic rule for a hormone must be this type of structure, but the general concept is once they get into the circulatory systems, they will interact with receptors. So if you have a cell, okay, and on that cell in the outer membrane is usually like receptors, okay? And if that cell is then bathed in like the extracellular matrix, the hemolymph, the hemocoel, or the blood, right? Hormones will be circulating around, and usually they'll like bind to these special receptors, okay? And then they usually get imported into the cell. And then oftentimes what happens is, just like Lauren said, they cause a signal cascade. That means stuff changes in the cell. And what's usually changing when a cell changes its behavior? What does usually that mean? If a cell changes its behavior, how is that process encoded, I guess? Which part of the central dogma do you think would be involved in changing cell behavior? Translation for sure, but more the impact is going to be on transcription. So hormones typically will get into the cell, and they'll usually interact with stuff that makes its way to the nucleus, and these things downstream usually turn on or off certain genes. So essentially it causes changes in transcription, which would be mediated by transcription factors turning things on and off. Okay, that's usually how hormones are working. So in the plant, there's two key hormones that are important for micropropagation. These are cytokinins and auxin. Okay, so these are two sort of, I guess, they're not flip sides of a coin, but they're kind of considered that they do kind of like opposite things. The cytokinins are growth hormones. So a cytokinin is going to stimulate, actually the term comes from cytokinesis, which is what?
It's when a cell splits.
The cell splits apart. That process is cytokinesis. So when a plant cell encounters cytokinins, it induces it to replicate and undergo cytokinesis. Okay, so that's how they're stimulating just kind of general growth. And auxins are sort of famous for stimulating root growth. And I should say the cytokinins are more famous for stimulating like bud or stem growth. So if you treat sort of like a little culture of plant tissue with cytokinins, you're going to start to see things start to sprout up. And if you treat them with auxins, you're going to sort of start to see things like root down, is sort of the general concept. And I'm simplifying it a little bit here. Usually you're mixing up ratios of cytokinins and auxins to do this behavior. But if you have a high concentration of cytokinin, you're going to get this kind of behavior. And if you have a high concentration of auxin, you're going to get this type of behavior. Okay? So the discovery of these plant hormones was very important for our ability to do this. And actually, I'll talk a little bit about the discovery of the first cytokinin because it's an interesting science story. When they were first sort of screening compounds, so what do I mean by that, screening compounds? If you have a plant cell, the experiment was they have a plant, they have some plant tissue, okay, in a petri dish, and they want to see how they can get it to grow. So they would start by just screening compounds. What does that mean experimentally?
They essentially were just using it just to see what would happen.
Yeah, they would essentially just add a bunch of stuff, like add whatever chemical they have around and see if they can use anything that can stimulate plant growth. And actually what they first did is they took herring sperm, so they took fish sperm, and they put the fish sperm on the plant cell. And for whatever reason, it started to sprout, okay? And then they analyzed the herring sperm, and there was a component in there that was essentially like mimicking cytokinin. And that chemical was, do I have it in here? I think it's kinetin, something like that. You don't have to know that name, but that's kind of how they originally sort of figured this out. It was just kind of random luck. And I like the story because you wouldn't necessarily think to like, oh, I'm going to take some fish sperm and put it on my plants and see what happens. So I like that story. Anyway, that worked, okay? And that was sort of this facilitated the discovery of cytokinins. Okay, so these are sort of set up as a budding versus rooting dichotomy, kind of, in simplest terms. Okay, so now with that knowledge, what do you do to micropropagate plant tissue? So there's 4 stages, okay? And the first stage is selection of the mother plant. So by that I mean like you were talking about, maybe you'd use this as a means of mass production, or maybe you'd use this as a means of selecting a particular phenotype. That's what I mean by selection of the mother plant. You have something that you've generated that you want to clone. And usually when they do this, usually they will grow the thing that they know about in a greenhouse beforehand, okay? And they'll use that thing that they grew in the greenhouse as the mother plant. Why would they do that?
It'll be healthy.
It's going to be healthy for sure, and they can sort of keep as much contaminants out as possible. The major issue with micropropagation of plants is it takes forever. It takes weeks, okay? And whenever you have something that's being cultured for weeks, you worry about contamination. That's a major issue. So oftentimes the thing that they're going to micropropagate, they won't just take that from the field. They won't just grab that from outside. It will have been sort of cleanly grown in a greenhouse to prevent as much contamination as possible. Okay. Then the next stage, I'm looking at this. I should say, before the next stage, they'll take the plant, okay, so if here's the plant, and then they can cut it up into little bits, and depending on the plant will depend on what tissue you're going to take or what tissue is going to be best for micropropagation. It's going to be different for every plant, okay? But they'll take those plants and they will surface sterilize. So they'll cut it up into little bits, they'll put it in bleach, and the function of that is then to kill essentially every single microorganism that's on the surface of that plant, so that the only thing they're introducing into the culture media is plant tissue and not contaminants. Okay, I guess step one is pretty big. Hang on. There's like subparts of step one. So subpart step 1A would be the selection. Subpart step B would be cut, surface, sterilize. Okay, and then subpart C would be they're going to actually put it in media. Okay, and the media is going to have some components in it. It's definitely going to have the cytokinins to stimulate sort of like some sprouting, okay, and some cell replication. And it's going to have some sugars, and it's going to have some salts, and usually it's going to have agar. So you're usually using, I guess, something that's just sweet and solid. And that's why we worry about contamination, because anything that's going to eat the sugar will contaminate that. Okay. And then what they'll do is once they get that to work, there's a stage called multiplication. That's just obvious. They just keep repeating this. They can just keep cycling back, repeating, until they've essentially got like a whole bunch of petri dishes with a whole bunch of little plant pieces. Okay? And as they sprout, you can take these out, you can cut them apart, and you can just continuously keep replicating these over and over and over until you have a bunch of them. My phone keeps shutting off. Okay. Now, the problem with these two stages in sort of plant biology is that these two stages, they're eating the sugar, okay, which means they're not taking in sunlight to photosynthesize. So the plant during these first two stages sort of, it doesn't lose its ability, but it gets used to sort of eating the sugar in the media. Okay, it's not used to producing its own. So there's a process where you sort of have to, between steps 2 and 3, you need to transition the plant to becoming able to do photosynthesis again and being able to sort of go out into the world and become an actual plant. So the third step is called rooting and acclimatization. Okay, and this step is now going to involve the auxins. So they'll add the auxins, okay? And now these little plants in the petri dishes will start to sort of like root out. Okay, so they're becoming like more like plant-like at this point. And the auxin is going to help them sort of go back and transition to a photosynthetic state. And it's going to sort of, the auxin also helps stimulate sort of all the things that a plant needs, in sort of, in a sense that it's going to make it's going to start, how do I want to say it? It's going to start the cell differentiation process to a point where it becomes more of an independent plant. I think that's the best way to say it. Okay, and then the step 4 is the final step. You can now take those plants and you can replant them in either a greenhouse or outside, and now they can generate independent plants all by themselves. Let me see if I mixed anything. That's the gist of micropropagation. Do you guys have any questions?
So you said step 4, they can now produce independent plants by themselves. So say if you had taken a hybrid that was sterile, you're
Yes.
saying it can now produce individual plants?
It can't reproduce, but that little tiny piece will regrow into a
Okay,
full
I see what you're saying.
full plant independently. But once that plant grows up, it will still be sterile.
Gotcha.
But it will generate all the leaves or the fruit or the roots that that particular plant has. Now, sometimes there's a term called, somatic embryogenesis. That's sort of what we talked about in the beginning. There's different flavors of plant micropropagation. Sometimes in certain plants you have to do it a certain way, and sometimes they'll take a tissue, and just like we talked about in the beginning, they'll add certain chemicals that actually make it become like a seed or a zygote. And then from that, they can sort of regrow. So that process is called somatic embryogenesis. That's the process of taking a somatic cell and making it go back to its totipotent or pluripotent state. What's the difference between pluripotent and totipotent? So I just threw out this term. We knew that this term means it can generate all possible cell types.
Pluri just one.
Well, pluri's 2, so it's, or 2 or more. So this is kind of like it can generate some, but not all. So you'll hear pluripotent stem cells. They can only return to a couple different cell types, whereas a totipotent stem cell could generate every single cell in that entire organism. Okay, that's essentially it. I will just say one final thing, which is grafting. Do you guys know anything about grafting?
When I read through this originally, so is it similar to skin grafting?
Skin graft.
Where you get a skin graft,
You mean
like transfer tissue?
Yes, except it's different in a sense that with grafting, you can actually take different strains, and in some cases, I think different species. Like you could have, say, some plants are really good at generating roots, okay, but they may not be very good at generating the fruit. So sometimes what I don't know if I'd say horticulturist. I'm not a plant person, so my terminology is probably terrible. But sometimes there are cases where you want to take the roots of one plant and then you can graft onto it the branches of a different plant, okay? And in that sense, maybe you could produce, they'll do this with grapes. So they'll take a certain grapevine that's really, really good and they can graft it. And then that upper graft is really, really good at producing lots of fruit, whereas maybe the roots are sort of pest resistant, or maybe they're really, really strong rooting systems. And the way you do this, you can just cut it and they'll literally just take tape and they'll tape these things together and they can grow together. So this is something that is I had never even heard of it. It's kind of unique to plants. So it's really interesting in that sense. So I'll just say that that's what that is. If you ever hear that, that's what they're doing. Any questions? All right, with that, that's the end.
So are you familiar with a skin graft, like I was saying earlier, where burn victims will get a piece of skin put onto them?
I've heard of that, and I know yes, I know about the procedure. I've never done it, but I know, and then I know the issue. One of the issues is rejection. Will they reject the skin graft? And there's some compatibility issues there. Why? What's the?
Well, no. This is kind of a side story. It's kind of funny, but when you sent this out and I read it, it's what is grafting? And I looked it up. My whole life I thought it was skin graft, pH. So
yeah,
that was an awakening moment for me. I went and looked into it for a minute.
No, they can do it with plants. That's the gist. All right. Did I actually record that? I think I did. Yes, I did. Okay.